And we are recording. I didn't even bother writing the stuff I usually write on there. A lot of it. Same old story, right? You can no longer retake exam 1 to change the grade, but you can take it for the practice. You can meet me online to get all the answers for exam 1 and 5% boost to the grade. And you're going to need those answers to study for the final exam. Exam 2, you can still retake, even if you've already retaken it. You can keep retaking it until exam 3 is due. Don't know when exam 3 is going to be due. In a perfect world, it would be due on April 19th-ish. But we'll see. It depends on how long it takes us to get through this material. And then once exam 2 closes, then we can meet online for the correct answers for exam 2. Again, in a 5% boost to your grade. And again, you can do that for any assignment. Meet me online, get the detailed feedback, and a 5% boost. Another thing I usually put up there that I didn't put this time is remember the officially late work and corrections are not accepted. That's what the syllabus says, just to cover my own rear end. But unofficially, I recommend that you turn in stuff that you've missed, make corrections. The only catch is my priority is new material. So if you turn stuff in late or you turn in corrections, I can't guarantee you I'll have time to grade it, but I guarantee I'll try. Obviously, because of the fact that I'm prioritizing newer work and because of the fact that other than that, grade stuff in the order in which it was received, the sooner you turn it in, the better. So there's still time to turn things around for those of you who are not doing too well. Any questions about that? All right. As I've been saying, we have lab today. It is, even though we're meeting in person, it's completely virtual, so you need to bring a computer. A few of you already did the lab. And just this morning, I went and saw, I haven't been having a chance to grade it yet, but if you've already done the lab, I've left you a comment that said you do not, I've at least looked it over and saw that you did the right thing. I don't know if you get it 100% or not, but anyway, I've already left you a comment if you do not need to come to lab today. So if you get that, check your labs to see if I left that comment. The rest of you, I will see you in lab today. Normally, I'd say don't be late, but since it's a virtual lab and it's not a team effort, yeah, I really don't care. I'm always picky about being late because I want you to, it's fair to your classmates to make sure you're on time. That way you're not coming in late, confused, and messing up everybody's work. So with individual efforts, I really don't care. But you still need to be here. Anyway, any questions about pre-lab today, or excuse me, lab today? All right, just as a reminder, next week's lab is also virtual, and in this case, it doesn't even have a pre-lab. Yeah, that's it. So just like this week's lab, if you go ahead and do next week's lab, there's a chance that you do not need to come to lab next week because again, next week's lab is 100% virtual. And Marley, what you just missed right before you walked in, I was saying some people, such as yourself, if you've already done the lab today, I've left a comment this morning and you do not need to come to lab today. You've already done the lab. Anyway, any questions about lab today or the pre-lab that is not due on Friday or next week's lab? All right. So as always, I'll remind you attendance is mandatory. Participation is graded. To get the points, you're going to send me an email that lists all the attendance words that I say throughout the video. If you're watching or throughout the lecture, if you're watching the video at a later date, also take a screenshot every time I say one of those attendance words and include the screenshot in the email. And include one question about the material covered in the lecture. And as I've been doing recently, if you are a person, then one of those questions, or excuse me, one of those words will be extra credit, right? So again, like I keep saying, if I give out 5 words today and you turn in 4, if you missed one, that's full credit. If I give out 5 words today and you turn in all 5, that's a little bit of extra credit. Help buffer your grade. Anyway, speaking of attendance, let's do it really quickly. Ashley, it's here. Barrett is here. Wooten, it's not here. Sheringham is not here. Dickens, not here. Clark, not here. Inca is here. Isaiah is not here. Copes, Smith is not here. Watson, not here. Haley is here. Lucas is here. Marley is here. Noah is here. Savannah is here. Mia is not here. Edwards is here. Lucas is not here. Trey man came in while I was talking. Did I miss anybody? Okay. So another thing, I don't say this enough either. This isn't a requirement, it's just a recommendation. If you're an athlete and you need me to fill out one of the progress reports, I recommend, don't require, I recommend emailing me the day before, like, hey, tomorrow I'm gonna need you to fill this out. Just for your own sake, because if you need it, like today, if you need one, you have to follow me into my office. I'm gonna have to run the report and do all that. So I have no problem doing it, but for your own sake, it won't hurt. Time is valuable. I mean, and I'm gonna be sitting in my office for office hours anyway, so it doesn't matter. Anyway, any questions about announcements or reminders? Does anybody else have any of your own announcements or reminders? Okay, then. Let's jump into it. Did you go? First word for attendance then is going to be manatee. Today is National Manatee Appreciation Day or National Manatee Day or something like that. Have any of you ever seen the manatee in the wild? Where was it? The Gulf. The Gulf? Like in Fort Myers?
More like Clearwater, St. Pete.
Okay.
Like right in Shoresbury.
Okay. Great. Did you get to get in the water with it at all?
I'm not allowed to touch it.
Oh, really?
It's legal.
Oops.
Well, I mean, it's like not like one of those things where it's like, oh, but I mean, they will fly you if they catch you.
I lived in I grew up in the Keys and used to live in the Mexican canals. And one day I was coming home from schools and they were going swimming. We got to swim with it. I don't know. I guess it may have broken the law. I don't know. Anyway, let's jump into it. So, you guys are already familiar with Punnett squares. The last thing we actually talked about on Monday was showed you this when we were talking about the law of segregation. We were talking about how your alleles segregate, right? So, for example, when this was crossbred, when this purple flower, which is heterozygous, goes through meiosis, those two alleles that it has segregate. So half of its gametes have a big P, half of its gametes have a small p. So yeah, and then we looked at the possible results, which is again a Punnett square. Now you've already been exposed to it there. You've already been exposed to it in lab, so probably right now I'm just telling you stuff you already know. So let's talk about it anyway. Punnett square highlights the 4 possible combination of gametes. And you should get familiar with them. You will need to do some of those for the exam. There will be basic questions on the exam, and I'll have scrap paper for you, but you will need pencils or whatever you want to work with. But anyway, there will be questions when I say this thing mates with that thing. What are the chances of their offspring being this? And yeah, and there's that worksheet that I shared with you. And if you need help finding it, let me know. It's great practice, and you'll get extra credit for doing it. So, yeah, you should look into that. Anyway, each square represents an equal probable product of fertilization. So, for example, again, the chances of having a big P is equal to the chances of having a little p, right? Everyone is equal. There are also not some exceptions, but we can expand on that in real shortly. But for now, I'll just leave it at that. Yeah. So, any questions about Punnett squares for now? We will come back and give some more examples. Okay. Let's talk about genetic alleles and homologous chromosomes. So this is stuff, again, that you already should be familiar with because of previous conversations and because of previous labs. Right? Labs are actually ahead of lecture right now. As you probably already know, but you definitely need to know for the exam, you need to know the difference between phenotype and genotype. Right? The phenotype, at least according to the textbook in this portion of the textbook, is the physical appearance, and the genotype is the genetic makeup that is responsible for that physical appearance. I would like to point out, though, I'm just writing down what the textbook said. A better definition of phenotype is not necessarily the physical appearance, but more like, I don't know, the phenotype is the result of the genotype. Because, again, sometimes your phenotype is not something you can see. As you're going to learn later, we're going to use sickle cell disease as an example for quite a few things. And that's a disease that is a phenotype because it's a result of a particular genotype. If you have this genotype and you have that phenotype, you have that disease, but that's something you cannot see. There's other things like wet earwax versus dry earwax. There's a genotype that is responsible for that. It dictates whether you have wet earwax or dry earwax. And again, that's something you really can't see. That's not a physical appearance thing. Anyway, so make sure you understand the relationship between phenotype and genotype. Any questions about those first 2 bullet points? All right. So we're again back up to where we're talking about. Remember, we're in the middle of this conversation about what Mendel did, Mendel being the father of genetics. And remember, I'm going off the old textbook, which presents this almost like a story of what he did, and that's what we're talking about now. He did all these studies where he was looking at characters, and to remind you the difference between a character and a trait. Like, I'll give you some examples. Character would be eye color. And then there's different versions of that, and those are called traits. So blue eyes would be a trait, green eyes would be a trait, brown eyes would be a trait. Chin dimple, or chin, right? The chin could be a character, chin dimple would be a character, and the trait would either be you have it or you don't. Those are just some examples. But anyway, as we've already mentioned at the very end of our conversation on Monday, when he did these crosses, he found out that the parental trait disappeared. At least one of the parental traits disappeared in that first generation, right? So you have the parent generation, right? The purebred mating with a different purebred, right? And then their offspring in that generation, there was one of those traits that always disappeared in that generation. And then he crossed that generation with itself, right? So then we have the next generation, and that trait would always reappear in about 75% of the, 25% of the offspring. So that's nothing new. Again, just to remind you, this is the slide we left off on Monday, right? So everything I just read to you is this right here, right? We cross two purebreds. That F1 generation always loses a trait. In this case, the trait was white flowers, right? 100% of the offspring of this mix were always purple. But then he crossed those, and then 25% of the time, they had white flowers. So that trait came back. And for us, again, that's probably not very interesting to you, but at the time, that must have been amazing. Like, wow, that trait did not disappear. Here we have just hundreds, thousands of purple flowers, and every time I cross them, I get 25% of them come out white. Anyway. Yeah, so you already know that. You need to know that. You'll get more familiar with it when you do some more Punnett squares. Some other stuff you need to know about, which you should already know, hopefully is a review. But the next I'm going to show you shows a pair of homologous chromosomes. And to remind you what a homologous chromosome is, you learned that when we talked about meiosis. Homologous chromosomes are chromosomes that have alleles of the same genes. And so you'll notice each chapter kind of describes homologous chromosomes in different ways. Last time, I think we said the homologous chromosomes are the same chromosome, but you have a different version from your mom versus your dad. Now, since we're talking about genetics, we're kind of focused in on the genetic aspect of the chromosomes, right? They have alleles of the same genes. Meaning, just as an example, I don't know where it is, but if the gene for chin dimples is at the very middle chromosome, or the middle of chromosome 5 in my genome, that means for every human who doesn't have something wrong with their chromosomes, the gene for chin dimple would be the very middle of chromosome 5 for all humans, right? That's how homologous chromosomes work. They have the same genes in the same location. Which brings us to a term called gene locus. So I keep saying the same genes in the same location. So there's a word for that location. It's called gene locus. That is the specific location of a gene on a chromosome. So again, you can think of any gene, any human gene, and look it up. And it will tell you where it is. Like, oh yeah, that's on the very tip of chromosome number 13. And for every human who doesn't have something wrong with their chromosomes, that would be the case, right? That, whatever gene I'm talking about, that hypothetical gene will be on the very tip of chromosome number 13 for every human. Which is what this bullet point here says, basically. The alleles of a gene reside in the same locus on homologous chromosomes. But of course, as we've already talked about, when we talk about these alleles, remember, an allele is just a different version of a gene. So even though we might have the same gene at the same location, all humans would have the same gene at the same location on the same chromosome, we might have different versions of it, and those versions are called alleles. And of course, sometimes we might have the same version of it. So sometimes they're same, which is homozygous. Sometimes they're different, which is heterozygous. So none of this is new, except maybe this word gene locus. And even the concept of the gene locus is not new. This might be the first time I've given you that term. Gene locus, the specific location. So the next slide is going to be a picture of what's on this slide. So you may have questions about this slide before I show you the picture. Okay. Here's a pair of homologous chromosomes. I don't know which one. It's a hypothetical one. Let's just call it chromosome number 8, whatever. It doesn't matter. Point is, you have a pair of homologous chromosomes. Say this one came from your mom. This one came from your dad. You're looking at these different genes. Whatever hypothetical gene P is, right? This person, you can see they're at the same location on the same chromosome. And in this particular case, it's a big P, right? It's homozygous dominant. Just so happens to be whatever gene that is. They got the same allele from their mom as they did their dad. Same over here. Whatever gene A is, it's located in the same position, right? On the same chromosome. And in this particular case, they both happen to be homozygous recessive, right? So they got the same version from their mom as they did from their dad. And in this case, gene B, this hypothetical gene, is located at the tip of this chromosome. And it's at the same place for both chromosomes, right? The one from the mom and the one from the dad. In this particular case, they got a dominant one from their mom, a recessive allele from their dad, and they are then heterozygous because they have one of each. Make sense? Now, to back it up a chapter, when this person goes through meiosis, right, those homologous chromosomes are going to separate. So half of their sperm are going to have, or gametes, excuse me, could be a male or a female. Half of their gametes are going to have a big B, half of their gametes are going to have a small b, right? So those things are going to be segregated. That's the law of segregation. But even though they're segregating, 100% of their gametes are going to have a small a, because that's all they have to offer. And 100% of their gametes are going to have a big P, because that's all they have to offer. But again, just to remind you, even though technically right now, I'm just trying to introduce you to this concept of a gene locus, or loci in plural. I still want to throw it back and remind you what the law of segregation is, right? These things will get segregated during meiosis. So any questions about this slide? All right, let's throw it back to something else we've already talked about. Law of independent assortment. And I mentioned this when we talked about meiosis, because I said in the first round of meiosis, when all those homologous chromosomes line up together, right? Chromosome number 2 from your mom, chromosome number 2 from your dad. Chromosome number 17 from your dad, chromosome number 17 from your mom. I don't know, chromosome number 1 from your dad, chromosome number 1 from your mom, whatever. It's all random, right? Now they're always together, right? Chromosome number 1 is always going to be with chromosome number 1, but they're all random the way they line up, right? It's not all mom on one side, dad on the other. That's the big picture. If you remember that, you should be okay. Now I'm going to add some more details. But before I do, let's talk about a dihybrid cross. We've already talked about a monohybrid cross. Remember, a monohybrid cross is what you look at when you look at those Punnett squares that we've looked at. Those 4 squares, we're looking at a crossing between 2 individuals, and we're only looking at one character, right? So we looked at, for example, in our lab, we looked at what happens when you cross somebody with a chin dimple with somebody who doesn't have a chin dimple. And dihybrid cross makes it a little bit more complicated, because now in that case, you're combining 2 characters at once and looking at it. Like, all right, what happens if you cross somebody who has a chin dimple and blue eyes with somebody who has no chin dimple and blue eyes? The bad news is that's actually, even though it's only adding 1 more character, it's a lot more complicated. That's the bad news. The good news is you will not have to do any dihybrid crosses for the exam. So any kind of genetics questions you get will be a monohybrid cross. When I say what happens when you cross this and this, what are the results, right? And when I do that, we're only going to be talking about 1 character. Now, granted, and I'll explain this a little bit later when we get to it, sometimes it'll be a 2-part question. Like I might say, what happens when what are the chances of this person and this person mating and having a son with this trait? So then you're looking at 2 things, and that makes the math a little bit more complicated, but not much, and we'll cross that bridge when we get there. But anyway, dihybrid cross, again. Now we're not looking at 1 character, we're looking at 2 characters. So remember, these things, these genes are on different chromosomes, right? So the gene, and I don't know this number, I'm making this up, but hypothetically, let's say the chin dimple gene is on chromosome number 5, and the eye gene is located on chromosome number 17. Those things are completely independent of each other, right? They're not inherited together, because you might have gotten like when you create your sperm, your eggs, your gametes, when they segregate, you might have a sperm or a gamete that has, I don't know, your mom's chin allele, but your dad's eye allele, right? They're completely independent of each other. But if they were not independent of each other, this is what we would see here. It's this first bullet point. This is not what happens. This is what would happen if this were the case. If they were inherited together, then that next generation, that F1 generation, would produce only the same two kinds of gametes they received from the parents. And the F2 would still show that 3-to-1 phenotype ratio. In other words, they would travel together. And again, like I said, that would mean if you got your mom's chin dimple gene or allele, then you would also get her eye color allele. But it doesn't work that way because they are on different chromosomes. Usually. And we'll come back to that usually idea. So there's no need to write this down right here. This bullet point we're talking about right here. Because this is what does not happen. This is saying if things happen that way, this will be the results. And that's not what happens. In other words, if things happen that way, everything I taught you would still be valid previously when we talked about those monohybrid crosses. But what we're saying, it doesn't happen that way. Instead, it happens like this. They are sorted independently. So now you have 4 different genotypes. So remember when we did a monohybrid cross, let's see if this thing will work. Yeah, I don't want to get into that. Won't get into that yet. Anyway, yeah, you can see here that there's 4 different genotypes, right? Well, I can do that. So if it was a monohybrid cross, right, you would either have, if we're looking at R, you would have big R, big R. Did you even do it that way? No, I don't want to make it. I'm going to make it more complicated if I do that. I'll hold off on that. I'll bring that up later. Excuse me. Anyway, so this is what happens, right? Now, instead, that first generation, that F1 generation now has 4 different genotypes. You can see, right? Again, we're looking at 2 characters. So this one has a dominant, recessive, dominant, recessive, and recessive, right? And that's just the gametes, right? Because remember, gametes only have 1 allele for each gene, which is why those genotypes are not, RR, YY, RR, YY. They're RY, 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 RY. Anyway, once you cross those, remember, because that's the F1 generation, once you cross those with each other, then the result is this. You now have 4 phenotypes in a ratio of 9 to 3 to 3 to 1. This may sound complicated, but for this 100 level class, let's put it this way. When you get a phenotype ratio of 3 to 1, you should think of a monohybrid cross. When you get a ratio of 9 to 3 to 3 to 1, you should think of a dihybrid cross. And more than likely, I'm not even going to ask questions about this on the exam because, in my opinion, it's a little bit more complicated than it should be for a 100 level course. But it is still important to understand, again, the inheritance patterns are different when you're looking at 2 characters instead of just 1 by themselves. Any questions about this before I show you a visualization of the words that are on the screen? All right, so here's what I'm talking about, using the pea plants as an example. On the left is what would happen if those traits were inherited together, but they're not because they're on separate chromosomes. So instead, this is what happens, right? So think about the Punnett square you did. That's it. That's just another Punnett square. But now instead of looking at 1 character, we're looking at 2 characters. You can see it's much more complicated. Imagine if you added 3 characters, and we will do that later. But anyway, again, to remind you, you would not have to do anything like this on the exam. The point here is, right, we have a purebred, yellow, smooth, right? So that's dominant. There's 2 traits, and it's purebred, and it has dominant for both. Green and wrinkly, recessive. So this is purebred recessive for both of those traits. When this thing goes through meiosis, those things segregate, right? But even though they segregate, the only thing it has to offer is a big R and a big Y. The only thing this has to offer is a little R and a little Y. Obviously, when those come together, you have 2 heterozygotes, right? Heterozygote for the R, heterozygote for the Y. Now this, right, when they segregate through meiosis, you can have 4 different options, right? You can have a big R and a big Y, those when they segregate, or you can have a little R and a big Y, or you could have a big R and a little Y, or you could have a little R and a little Y. Again, just try to picture where all these things are getting segregated in meiosis, and they're not traveling together because they're on different chromosomes. So those are all your different options. That would be a lot more complicated, right? If you had to do that on the exam. I said, all right, what are the chances of these things mating and the plant having an offspring that has a heterozygous genotype, right? You have to do all this and count them up, and you have this 1, 2, 3, 4 of them. It'd be a lot more complicated, but again, you don't have to do that. Any questions about this slide? I know it seems a little bit complicated, but again, I'm not going to get that complicated on the exam. So a dihybrid cross supports Mendel's hypothesis that each pair of alleles segregates independent of the other pair during gamete formation. Again, in other words, looking at that dihybrid cross, which is what he did, that's how he came to this conclusion. Because I already told you what independent assortment was. But then I kind of backed up and said, and he's the person who figured it out. And now I'm backing it up again and saying, that's how he figured it out. I did a Quentin Tarantino on that, right? So I gave you the information, and we backpedaled. Learned how we got there. So again, when we're talking about meiosis, the way we describe independent assortment was that it's not all the mom chromosomes on one side and one dad chromosomes on the other. And that is still true. But now that we're talking about genetics, we're still talking about the same independent assortment, but we're focusing on a different aspect of it. So when you're only worried about the genes, then we say the inheritance of one character has no effect on the inheritance of another. Another good example, I don't want to muddy the waters yet. We'll just leave it at that for now. Anyway, that is Mendel's law of independent assortment, which is that independent assortment is seen in inheritance of two characters. That above is the law of independent assortment. Anyway, the next slide I'm going to show you is going to give you some examples, some more examples of independent assortment that aren't plants. They're going to be dogs. And they're just examples, so you don't need to write anything down. I'm not going to test you on anything about these dogs. But before I move forward, does anybody have questions about this slide? Okay, so I want you to understand, before I move forward again, I just want you to understand what the law of independent assortment is. Not necessarily the definition. You definitely need to understand it because there will be a question where I either explain it or I do give the definition or some version of the definition. So again, memorizing the words verbatim won't help you. Understand the concept, right? These chromosomes line up independent of each other. And they don't, if you have different genes on different chromosomes, therefore they don't travel together necessarily. Just because you've got your dad, I don't know, it's because you're giving your mom's chromosome number 1, doesn't mean you're also going to give your mom's chromosome number 2 when you create a gamete. You might give your dad's chromosome number 2. And there's different genes on both of those chromosomes. Anyway, if there's no questions, here's some pictures I was talking about. This is from the old textbook. So here we have two different phenotypes and genotypes. For Labrador retrievers, being black is dominant, being brown is recessive. Being able to see is the dominant trait. Being blind is recessive. So when you look at it, these are the different possibilities, right? You can have this one's completely dominant, right? You can see and it's black. This one has the dominant color, but the recessive eye condition, right? It's blind. This one has the recessive color, but the dominant eye condition. This one's just completely recessive. Some interesting things to note, and this is going to come back here shortly. If you look at this, think about this logically, right? This is a little bit like what we did, in lab, right? So when you filled out that chart and it said, what's your phenotype and what's your genotype? Whenever you had a dominant genotype, you had to write both possibilities, right? So if you had a chin dimple, for example, you literally had to write big D, the word or, and then big D, little d, right? Because both of those are possibilities. Another way of writing that would be what we're seeing here. Like in this case, what I could have had you done, but I didn't want you to do that because I wanted you to become familiar with heterozygous and homozygous dominant. You could have written big D blank. Because in a sense, that's what we know your genotype is, right? If you have a chin dimple, that means it's dominant. That means we know you have at least one dominant allele, and the other one we don't know what it is. Could be dominant, could be recessive, we don't know because either way, you're still going to have the chin dimple. Does that make sense? So that's what these blanks represent. We know because this dog has a dominant color it's a big B and something, right? We don't know if it's big B, or big B, small b. We know because they can see it must have at least one one dominant allele, big N. We don't know what the other one is. Could be dominant, could be recessive. We don't know because it's a dominant condition. This one's a lot easier. For it to be black or brown, brown, yeah, for it to be brown, it would have to be homozygous recessive because that's how recessive traits work, right? 100% of your alleles have to be recessive for you to have the recessive trait. Therefore, it has to be little b to be brown. Same with its eye color, with its vision, right? For it to be blind, it has to be recessive. Little n, little n. 100% of the alleles have to be recessive for you to have the recessive genotype. So this concept's going to come back soon, but not quite yet. So just keep that in the back of your mind. Any questions about this slide? I'm going to put a big old X through it before we move forward. Just as a visual reminder that this is an example. I'm not going to ask you any questions about dogs on the exam. Anyway, bringing this back to a dihybrid cross, if you were to take a homozygous dog and cross it with a homozygous dog, so meaning if you were to take big B, little b dog, cross with a big N, little n dog, right? So they're both heterozygous for both characters, this would be the results. So really what I'm showing you here is another dihybrid cross, but instead of having a Punnett square with numbers and P's, you can see the results in these dogs, right? If you were to cross that, in theory, you should have 9 black dogs that can see, 3 black dogs that can't see, 3 brown dogs that can see, and 1 brown dog that can't see. It's the same ratio as we looked at before. It's just shown to you a different way. Now, of course, think about it like this too. If you cross 2 dogs, I'm not a dog expert, but I don't think they had that many puppies at once, which is a good time for me to introduce this concept to you, this idea. These are all just numbers. When we're talking about this kind of stuff, and we'll talk about this officially later, but what we're really talking about, the laws of probability, right? This is statistics. So in theory, again, when these 2 dogs mate, they're not going to have that many puppies at once. What that's saying is if those 2 dogs mate and keep mating and do it a few times, that if you were to take all the dogs, however many they had, then the ratio would be 9 to 3 to 3 to 1. About that, right? And it could be a little bit different, right? These are just the rules of probability, right? So after doing this, it might actually be something like 8 to 4 to 3 to 1, right? Or I don't know, 7 to 4 to 4 to 1. It might be slightly different, but that's what we expect. This is what we expect. This is the rules of probability. And that's an important concept to keep in your mind too, right? This is not straightforward. This is not what's guaranteed to happen. This is like, as you're going to see in the exam, the questions are worded, what are the chances of this? Not like what's definitely going to happen. That's why generally the question won't say, if this person mates with this person, how many of their kids will be this? And you can almost say it like that because, 3 out of 4 kids, whatever. But the more proper way to look at it is, what are the chances of this? Anyway, that being said, let's go back to this concept right here really quickly. Let's look at this dog. Again, we know its phenotypes. It has 2 dominant phenotypes. It's black, that's dominant. It can see, that's dominant. But let's say we got it from the shelter, so we don't know anything about its heritage, its pedigree. So we don't know its genotype, right? We know it's big B something, big N something. That brings us to this. Using a test cross to determine an unknown genotype. And a test cross, and I might ask you this, so maybe take a note of that you might need to know this for the exam. A test cross is a mating between an individual with a dominant phenotype, but unknown genotype, right? Because that's what we're trying to figure out, and a homozygous recessive individual. And you'll see why we do that once I show you. Long story short, again, the purpose of this test cross is to figure out that unknown genotype, right? That's what we're trying to do, is figure out this unknown genotype. So we can't cross an unknown genotype with another unknown genotype, because then we're just going to be even more unknown, right? That's not going to help us. But we cross something that is showing a recessive phenotype. We know its genotype. If you are recessive, if you show the recessive phenotype, then your genotype has to be 100% recessive alleles. So because we know that, then we can do this test cross and figure out the the genotype. Then we're going to use dogs again as an example, but obviously you can do this with anything. Not ethically. You couldn't do this ethically with anything. Like, for example, if you're trying to figure out your buddy's genotype, you can't like have them mate with that girl and that girl and that girl. Like, okay, let's look at their kids and figure out your genotype, right? That's not ethical. But you can do it with certain animals and plants, fungi, or whatever. Anyway, let's look at some examples. And these are, again, just examples. I'm not going to ask you any questions about this on example. Here we have this dog. We don't know its genotype. We're just looking at coat color right now. We're not even looking at vision. It's black. That's dominant. So we know it's big B something. We don't know if it's big B, or big B, little b. This dog, on the other hand, being brown, we know 100% of its alleles must be recessive. So little b, little b. So we look at two different genotypes, or two different possibilities, right? If this dog was big B, and mated with that dog, then all of the offspring we would expect to be black. Now, if this dog was heterozygous and mated with that dog, then this is what we would expect. About half of them to be black and half of them to be brown. Now, here's where it gets a little bit trickier. And I don't think I'm going to get this tricky on the exam. But yeah, this is just rules of probability. This is none of this is set in stone. So it is possible in this mating, right? We expect one to one, right? So if it has 4 puppies, we expect 2 of them to be black and 2 of them to be brown. Does that make sense? If it has, I don't know, 6 puppies, we would expect 3 to be black, 3 to be chocolate. Hope that makes sense. But again, the point here is that's what we expect. Technically, in this mating, it could happen that all of the dogs come out black. And for that matter, all the puppies could come out brown. But my point is, if you did this cross and 100% of the puppies were black, that suggests that this dog is homozygous dominant, but that doesn't prove it, right? Because it is still technically possible for this to always be the pair, right? Because we're just talking about these are 2 different sperms, right? There's a sperm with big B and a sperm with a little b. It is possible that only the big B sperms are the ones that do the fertilization. So it is possible that 100% of them come out black. Not likely, not probable, but possible. So keep that in mind. Not for the exam, just for critical thinking, right? But that being said, if you ever cross these, if there's any browns, that's 100% proof that this dog is heterozygous. Because with a brown dog, there is no other way to have a brown dog unless that black dog is heterozygous. I hope that makes sense. Academically, again, that's not very important because I'm not going to get that deep on the exams. But I want you to be critical thinkers. Like if somebody were to bring this to you and say, okay, you were to see this situation, like all 3 puppies were black. That proves that that dog is, the black dog, the black parent was big B, big B. Like, that doesn't prove it. It just supports it. Anyway, that brings us to this next concept. I keep saying, right, this is just the rules of probability. This is just statistics. None of it is set in stone. So let's talk about the rules of probability. Genetic crosses. Sorry, let me back up really quickly. Make sure I didn't skip something. I did skip something. Yep. Sorry. The next word for attendance is NB. Just the letters NB. I don't know if you can read that. The Norman Borlaug. Today is his birthday. And again, not on this email that you're sending for attendance, but if you want to look it up later, you can look up who that man is. It's relevant to biology, more so agriculture, but agriculture is a type of biology. Anyway, move back where I was. Sorry about that. Rules of probability. Here we go. Genetic crosses obey the rules of probability. And these are the same rules that apply when you're tossing a coin, rolling a dice, drawing cards, etc. Actually, to be more accurate, rules of probability they are the same. But tossing coins is the best example. Because when you're tossing coins, how many options are there? When I flip a coin, it's going to land on something, right? So how many different choices are there when I flip a coin? 2, right? Same thing with when we're talking about making gametes. You have 2 alleles, right? But that might be the same if you're homozygous, or they might be different if you're heterozygous. But the chances of this gamete being involved in the fertilization versus that gamete being involved in fertilization, it's 50-50, right? It's just like tossing a coin. Which brings us to, this is where it's a slightly more complicated, the rules of multiplication. But I'm not going to ask you the rules of multiplication, so there's going to be at least one question where you need to use the rule of multiplication. And it states that the probability of a compound event is the product of separate probabilities of independent events. The explanation is more complicated than how it works. I'll give you an example. I guess I'll use the same letter, big D, right? So if somebody has the chin dimple and they're heterozygous, they're big D, little d, right? So what are the chances, if you were to look at that person's gametes, what are the chances that you would pick a gamete that has a big D versus a small d, right? You look at all that, you just randomly pick they have a sperm sample and you just randomly pick your one sperm. What are the chances of that one sperm being a big D as opposed to a small d? Don't overthink it. 50%, right? Another way of writing that is 1/2, right? 1/2 chances of that being, yeah. Now that's the male, right? So what are the chances of the female? All right, big D here. What are the chances of us looking at that female's, all of her eggs and just randomly picking one and it happens to, again, be a big D as opposed to a small d? What are the chances of that? Don't overthink it because whoever said the last one was correct. 50%, right? You're either going to get a big D or a small d. So the chances of that person giving off a big D would be 1/2. So this is what I mean. This is what this bullet point, this complicated bullet point says. It's just this simple. So the chances of this person and this person creating a child that's